Okay, now suppose these are two wind, layer. Suppose, now listen, these two layers, suppose this is one layer and this is second layer. Now listen, this is the first layer, this is second layer. Now listen with all your attention. This first layer will move with much more ease with respect to the second layer, right? That means in this case this is also happening, that is when the last layer is moving, it is actually interacting with this static layer. That means the velocity of this, the velocity head of this particular fluid layer will be much less than that of the upper. That means suppose this is V1, this is V2, V3, V4, V5, we can obviously say V5 is greater than V1, right? The velocity head of layer 5 is greater than V1, right? Now listen, in this case, now we'll actually use this analogy to explain this. Okay, now listen, whenever we are actually filling the sheath fluid compartment, what is happening is this sheath fluid actually, what is the exact composition of sheath fluid, we'll talk about this in details later. Okay, why we use this sheath fluid, and most important is this, what is the exact composition of sheath fluid, we'll talk about this in details later. Now listen, now in this sheath fluid, what is happening is, suppose this is the third layer, this is the first layer, this is second layer, and this is third layer. Okay, now listen, just understand this very basic thing, that is, suppose this layer, this is the first layer, is interacting with this sheath fluid compartment wall, and this third layer is actually interacting with sample nozzle compartment, right? Now listen with all your attention, that is, suppose these are sheath fluid particles. Suppose these are sheath fluid particles. Okay, now you should understand that when this layer, the third layer of sheath fluid is moving, it is moving at a lower velocity than the second layer. Now listen, now this third layer is moving at a lower velocity and this first layer is moving at a lower velocity, but the second layer is moving at a higher velocity. That is a basic thing you need to understand. Now very important part here is that when these layers are moving, okay, these layers are moving, you should understand this now, very basic thing. When these layers are moving, now, something called Reynolds number? Something called Reynolds number? What is the utility of this Reynolds number? Reynolds number will determine which layer will be turbulent and which layer will be laminar. Which layer will be turbulent and which layer will be laminar. Now listen, very important part you should understand here is turbulent. What do you mean by the term turbulent? How we define the term turbulent? Now listen, we should understand here the Reynolds equation, that is what? Rho Vd divided by mu. Here rho is the density of the fluid and mu is the viscosity of the fluid. Okay, now listen, if after calculation Re, that is Reynolds number, is less than 2300, then it is called laminar flow. Laminar flow. This is called laminar flow. If after calculation of this, if after the calculation, if Reynolds number is greater than 2300, then it is called turbulent flow. Now you need to understand one thing, that this layer, the third layer, was actually in a static condition when it is attached to this nozzle. Now listen, due to the drag of this second layer, this third layer is actually coming into acceleration, right? But you should understand, suppose you are standing still and I am running, okay, and we are both tied by a string. Suppose I am running at a faster rate, but you are actually standing still. When I am running, obviously I will try to drag you, right? But will you actually accelerate with respect to my own acceleration? That means, will you have the same acceleration? No, right? Will you have the same acceleration? Never, right? Why? Because you don't have the will to run, right? That is the basic reason you should understand here, that this layer wants to get attached to this particular nozzle, and this layer is actually a runner. This layer is actually dragging this layer, this second layer is actually dragging the third layer. Now listen, due to this drag, the third layer particles will actually fall with a uniform velocity, and that's why we'll create a laminar flow. Due to the drag of this second layer, the third layer will form a laminar flow, because its velocity is not that much great, right? Not that much big with respect to the second layer. Now listen, that means what we are doing is, in this setup, what we are doing is, we are actually creating a drag. Okay, we are actually creating a drag using sheath fluid, due to which there is a turbulent flow here. Sorry, there is a laminar flow here. Now listen with all your attention. Due to this laminar flow, we will immediately see the effect of this laminar flow. That is, the cells which were coming like this, due to this uniform drag, one by one they will come out of this system, one by one. Okay, they will come out of this system one by one. That is, due to this laminar flow, one by one these cells will come out of the flow cytometer, one by one, one at a time. Now listen, this process, this whole thing, this whole fluidic system, this whole function of the fluidic system, that is creating a laminar flow so that cells can come one by one, this whole thing is called hydrodynamic focusing. This whole thing is called hydrodynamic focusing. That is, we are focusing the cell, okay, we are focusing the cell in a laser system with the help of water, which is moving, dynamic. Okay, that's why it is called hydrodynamic focusing. This is called hydrodynamic focusing. Clear? Okay.